Chapter 5, Part 3. So we're going to talk about the multiplication cycles in animal viruses. So those are viruses that infect animal cells. And we'll talk a little bit later about the multiplication cycles of viruses that infect bacteria. So please try to separate that idea. So right now we're talking about animal viruses, viruses that infect animals, or animal cells rather. So general phases of the animal viral replication cycle. So a lot of this is tongue twisting, so just bear with me here. So I really don't have a mouthful of marbles. There's 6 steps, 6 phases: adsorption, penetration, uncoating, synthesis, assembly, and release. So the length of the replication cycle varies from 8 hours to about 36 hours, depending on which one we're talking about. So we're going to talk about each of these individually moving forward. So first we're going to talk about adsorption. Okay, it's not absorption. It's not absorption, it's adsorption. So a virus can invade its host cell only through making an exact fit, like a lock and key, with the specific host molecule. Has to do with that docking of the boat thing. So the host range is something that refers to the limited range of organisms that the virus can infect. So, there's viruses out there that can infect humans but can't infect cats. There's viruses out there that can infect dogs but can't infect humans, or birds, or trees, or whatever. So, of course, we're talking about animals here, so don't let me throw you off with that tree thing, but I think you probably may pick up what I'm saying. So when we're talking about host range, it means that, hepatitis B can attack the liver cells of humans, poliovirus, intestinal nerve cells of primates, rabies, various cells of all mammals. So some have more versatility than others. Cells that lack compatible virus receptors, okay, so the docking stations for those viruses, are resistant to adsorption and invasion by that virus. So if we do not have the proper receptors on the cell, if the cell does not contain or have the needed receptors that virus needs to dock and connect with and adsorb to, then that virus cannot invade that cell. So terminology, tropism, tissue tropism, is the specificities of a virus for certain tissues or cells. So, I think I mentioned earlier that the flu virus is not going to infect my skin, right? The flu virus has to be in my respiratory tract somewhere in order to infect me. The COVID virus needs to be deep in the lungs to infect you with that virus. So it's definitely interesting the way all this works. So I kind of blew this up and stretched it out a little bit because I wanted you to be able to see this a little bit better, but you can see there's a little line pointing to the receptors. Okay, so these would be like the receptors on the cell. And these spikes coming off of the virus, they're a perfect match to those receptors. So that virus can invade that cell then. If this was not a perfect match, then that virus is not going to be able to invade that cell. Okay, so after adsorption comes penetration and uncoating. So the flexible cell membrane of the host is penetrated by the whole virus or its nucleic acid. Penetration happens through endocytosis, when an entire virus is engulfed by the cell and enclosed into a vacuole or vesicle. So we think about what endocytosis is. We learned about that in Anatomy 1. So think about how that cell kind of just opens up and just kind of engulfs what's sticking to its surface. That's what's happening during endocytosis, during the entrance of a virus into a cell. There's also direct fusion. of the viral envelope with the host membrane. And there's a picture coming up next that illustrates both the penetration through endocytosis as well as direct fusion. So during direct fusion, the envelope merges directly with the cell membrane, liberating the nucleocapsid into the cell's interior. And you can see here, this is through endocytosis. So these little arms, these little lips like arms come out and kind of just like, whoop, I like what I see. I'm going to just suck it in, right? And then it just, the membrane comes with it inside the cell. And then the cell, the animal cell has enzymes that will basically break down, uncoat the virus, and that allows for the genetic material to be released into the cell, and that genetic material is hanging out there, and basically the ribosomes in our cells are like, whoop, whoop, yeah, hey, there's some DNA, I need to copy it, because that's my job. And so, or the mRNA does. And so it copies it, and when it copies that genetic information, that is the genetic information from the virus. Our cells are like these machines that just, it's either black and white. There's no in between. It can't decipher. It doesn't decipher that this is a bad guy. Its initial encounter anyway. And so it follows this recipe to make more viruses. And the process can be very damaging and can make you very sick depending on the virus. So over here is the direct fusion example. I don't know why they don't have these labeled, but the direct fusion is basically they have the receptors on the outside of the cell, and this likes something over here, otherwise the cell wouldn't take it in, but it's got a specific attachment, and then it makes this complex with the receptors, or the spikes on the virus rather. The spikes on the virus make this complex with the receptors on the cell, and that basically is like a fusion of the virus to the cell, and then it just basically dumps the virus into the cell. So those are the two examples of how they penetrate animal cells. All right, so next comes synthesis. And what is synthesis but replication and protein production? So we're if you go up here and look at this, we have adsorption, penetration, uncoating, and synthesis. So that's where we're at now. So in DNA viruses, they enter the host cell's nucleus and are replicated and assembled there. In RNA viruses, they replicate and assemble in the cytoplasm. So, you revert back to what we know about the COVID-19, that is an RNA virus. Everybody, I've heard all sorts of things about you're messing with our DNA with these, vaccines, and I can understand concern at how swiftly everything's moved, but we've never experienced a global pandemic before, so there's things that there was no corners that were cut. It was more like testing of this was simultaneous rather than in a linear fashion. RNA viruses are replicated and assembled in the cytoplasm. So the genetic or the vaccine never gets near our nucleus of our cells or our DNA. But anyway, we'll get into that when we get into the later chapters, because we'll learn all about that. I want to get too ahead of myself here. All right, so let's see. Okay, sorry, I had to gather my thoughts for a second. So we're in the synthesis phase of this, right? So the early phase of synthesis is the viral DNA enters the nucleus, and this is when we're talking about DNA, or a DNA virus. The DNA enters the nucleus of the cell where genes are transcribed into a messenger RNA, and then RNA transcript moves into the cytoplasm. to be translated into the viral proteins. Some enzymes might be needed to replicate the viral DNA. The host cell's DNA polymerase is involved in this phase. In the late phase, parts of the viral genome are transcribed and translated onto proteins, or into proteins rather, required to form the capsid and other structures, and new viral genomes and capsids are assembled, and then the mature viruses are released by budding or cell disintegration. So this kind of gets a little ahead of itself with the assembly and the release. So basically it talked about assembly and release right here, right? So assembly is the virus is put together using the parts that are manufactured during the synthesis process. So release is when the viruses are released by the infected cells. And this depends on how big the virus is, the health of the host cell. Some viruses can release 3,000 to 4,000 virions. Polio virus can actually release 100,000 virions. So you can imagine if there's one cell releasing 100,000 virions, the potential for proliferation or multiplication of this virus is very, very high. All right, so one is kind of a drawing or illustration, and this is an actual image. So this is a mammal cell, and then these are viruses, budding off in search of other cells to infect. And you can see here's some pictures here too. So good illustrations. So kind of a summary of what we've talked about. Adsorption, the virus encounters a susceptible host, adsorbs specifically to receptor sites on the cell membrane. The membrane receptors that viruses attach to are usually proteins that the cell requires for its normal function. Glycoprotein spikes on the envelope or the capsid of the naked viruses bind to those receptors. Penetration and uncoating, the entire virus is engulfed, endocytosed by the cell, and enclosed in a vacuole or vesicle. When the enzymes in the vacuole dissolve the envelope and capsid, the virus is said to be uncoated, and the process that releases the viral nucleic acid into the cytoplasm. Synthesis. Now, I'm not going to read all this, but basically synthesis is when all the parts of the virus are made. So, you're building, I don't know, you're making cakes, right? You gotta make sure that you make all the layers of the cake first, and then you gotta make your icing, but it all doesn't, just get put together. You have to take your time and put the cake together, which is what happens during the assembly process. So first you have to make all the parts, and then you have to put it together. Assembly is when the virus particles are made. Right? There's parts there and they're all put together. And then the release is when the cell actually either bursts due to being overfilled with viruses or they bud off. So damage to the host cell. Cytopathic effects, CPEs, are virus-induced damage to the cell that alters its microscopic appearance. This includes serious changes in shape and size to the cell. There can be intracellular changes. There can be inclusion bodies, kind of trashing up the cell. I looked this up and how to say it. I always forget how to say this. Syncytia. I'm saying that completely wrong. Syncytia. Anyway, fusion of multiple damaged host cells into a single large cell containing multiple nuclei. They're referred to as giant cells. The accumulated damage from a virus usually kills the infected host cells. So we'll stop here and we will pick up on the next installment of Chapter 5.